Hello, welcome to Chapter 5. In this chapter we're going to be talking about mutation and genetic variation. We're going to see how we can measure that variation between individuals and just briefly sort of talk about where that all comes from. So there are really three kinds of variation that we can talk about. We can talk about genetic variation, we can talk about environmental variation, and then we can talk about one that kind of combines the two. This is the genotype by environment variation. The best way to illustrate this is with an example. That example is the variation in skin color among humans. For example, you may look at two newborn babies and you might see that at time 0 they have different colors, and that would most likely be due to the different alleles at the genes that are contributing to skin color. That would be genetic variation. You might also have let's say identical twins, and let's say these identical twins were in college and one of the twins spends the whole spring break in the library. The other twin spends spring break down in the Caribbean. In this case, at the end of the spring semester, they would also have very different colored skin, right? And that's due to the environment that they were exposed of, the library versus the Caribbean. The last one is so then this would be environmental variation. The last one is genotype by environment variation, and this occurs when, so you might imagine two people that have roughly the same kind of skin color. They both go to the beach together. At the end, one is all lobstered out, completely red, totally burned, and the other one just tans. This would be due to genotype by environment variation, that the one individual has other genes that make it so that she tans, whereas the other individual that burns have genes that doesn't necessarily lead to that. So the two main objectives for this chapter is, one, to look at the mechanisms that are responsible for creating new versions of genes or alleles, or even in creating entirely new genes. And then we're also going to, at the end, look at how biologists quantify or measure the amount of genetic variation in a population. All of these things obviously are very, very important for evolution via natural selection, because it's that genetic variation that natural selection works on to produce changes in an evolving population. So before we talk about genetic variation and look at where new alleles come from, we should probably do a quick review of DNA. So DNA is a nucleic acid. A nucleic acid is a polymer made up of monomers. The monomers that make up DNA are called nucleotides. And here you have a figure of a nucleotide. Each nucleotide is made up of 3 different parts: a phosphate group, a carbon group or a sugar group, and a nitrogen containing base. If you look at the carbon there in the middle, you will notice that each of the carbons within the sugar is labeled. You have 1', 2', 3', 4', and 5'. In DNA, we're particularly concerned with the 5' carbon and the 3' carbon, especially when we start, if you remember back in intro biology classes, when we talk about DNA replication, we concentrate on what's going on at the 5' carbon and the 3' carbon. The phosphate group is connected to the 5' carbon. The nitrogen containing base is connected or bound to the 1' carbon. And then the 2' carbon is also important, but mostly important in distinguishing between DNA and RNA. In DNA, that 2' carbon has a hydrogen bound to it. In RNA, it has a hydroxyl group. If you remember, hydroxyl groups are made up of a hydroxyl group is made up of an oxygen and a hydrogen, and so DNA has one less oxygen than RNA. That's where the deoxy part of deoxyribonucleic acid, DNA, comes from. So a more in-depth look at the nitrogen-containing bases show that in DNA they come in basically 4 flavors or 4 varieties: adenine, guanine, cytosine, and thymine. Just looking at all 4, you notice that you can group them into 2 different groups. One group where you have just a single ring and another group where you have 2 rings. The 2-ring groups, or the 2-ring nitrogen-containing bases, adenine and guanine, are known as purines, and the single ring bases, cytosine and thymine, are known as pyrimidines. I'm not that concerned about you all knowing the details of these the detailed sort of chemical structures of the different nitrogen-containing bases, but I do want you to sort of understand overall general characteristics of the purines and the pyrimidines. The large ones are the purines, the smaller ones are the pyrimidines. And when we get to the binding between the nitrogen-containing bases, you will see that a pyrimidine binds with a purine and the purines bind with a pyrimidine. And here on the right is what that looks like. So you see thymine is bound to adenine. The type of bonds that form between the nitrogen-containing bases are hydrogen bonds. If you remember, hydrogen bonds are not exactly the strongest bonds, but if you have 2 or 3 hydrogen bonds forming between each nitrogen-containing base in the structure of DNA, which we'll get into here in a second, it actually lots of not very strong bonds result in a strongly bonded molecule if you have lots and lots of those bonds. Remember, thymine is a pyrimidine, adenine is a purine. You have a single ring base bonding with a double ring base. Also notice that when adenine and thymine bond with each other, it forms 2 hydrogen bondings. If you look below that at guanine and cytosine, you see that 3 hydrogen bonds are actually formed. Therefore, a DNA molecule with more G's and C's in it is going to be more strongly bound. The 2 different strands of the molecule are going to be more strongly bound than a DNA molecule with the majority of the nucleotides being adenine and thymine. If you look over on the left-hand side, you see that each nucleotide is bound to another through what we call a phosphodiester bond. This phosphodiester bond forms between the 5' phosphate group, or the phosphate group attached to the 5' carbon, and the hydroxyl that is attached to the 3' carbon. And so that bond forms, that links one nucleotide to the other nucleotide. There on the left, though, you see that it's a single-stranded molecule. Okay, so you have a single-stranded molecule. The backbone of that single-stranded molecule is formed by that phosphodiester bond that is forming between the sugar and then the phosphate, the sugar and a phosphate, sugar and a phosphate, with, on the other side, the nitrogen-containing base being attached to the 1' carbon. So if you get 2 single strands, right, you have complementary base pairs on the second strand. Those 2 single strands will form a double-stranded molecule, which you see there in the middle. And so you'll notice, again, review, that adenine always bonds with thymine and guanine always binds with cytosine, and the reverse is also true. And so once you know the code of one side of the molecule, you're automatically going to know the code of the other side. Also remember the opposing orientation of the strands. You have on one strand, you will go from 5' to 3'. Again, that's that 5' carbon end to the 3' carbon end. On the other strand, it's flipped, and you can see that there in the middle. You can see that it's then 3' to 5' on the other side. That is just the general orientation or polarity of the DNA molecule. So for DNA to replicate, all that needs to happen is you have to unwind or separate out those two different strands from each other, expose those strands, which now serve as a template, to the ingredients that are necessary to build that complementary strand, which are basically nucleotides and then some enzymes. Expose those strands or those templates to a whole bunch of nucleotides. Those nucleotides will go and bond to the complementary nucleotide. So for instance, A's will bond with T's, G's will bond with C's, and then in the end you just have to form those phosphodiester bonds between those newly incorporated nucleotides, and at the end you go from a parental strand of DNA to two daughter strands of DNA, which theoretically should be identical. I say theoretically because when they are not identical, as you can see happening there on the daughter strands on the right, when they're not identical, or where you have had a nucleotide, for instance, inserted where it shouldn't be inserted, that's when you get mutations. And essentially that's all mutations are, these changes in the DNA sequence, in the nucleotide sequence that compose a DNA molecule. So this is especially important when we're talking about DNA replication that is going on in our germline, in the cells that will eventually form our gametes, because then these get passed down to these mutations, then get passed down to the next generation, and they become the sort of variation that natural selection has to work with to enable populations and lineages to evolve. So to understand mutations, or in other words, a change in that genetic code, we need to also understand the flow of information. So the central dogma of genetics is that information flows from DNA to mRNA, and then from mRNA to protein. The process of going from DNA to mRNA is transcription. The process of going from mRNA, or that information converted from mRNA to protein, is what we call translation. And again, just as a refresher, the bits of information come to us sort of packaged in these things that we call codons. Codons consist of 3 nucleotides. Those 3 nucleotides, for instance, here in the piece of DNA that we're looking at all the way to the left, those 3 nucleotides might be CAA. Those are going to, when the double strands of our DNA molecule are separated, the gene's located on one of those strands, and that bit of information is going to be transcribed into an mRNA molecule. And in the same way that DNA goes through DNA replication, through transcription, we're going to produce this RNA transcript. And so your C is going to bind with a G, and now because it's RNA, and RNA does not contain G's but rather U's, which stands for uracil, the U's are going to bond with the A, and so the first codon, right, becomes GUU. That codon is then going to bond with an anticodon, and that anticodon within a molecule that we call tRNA is going to have at one end the anticodon, at the other end it's going to be bound to a specific amino acid. In this case it is valine, and that's how that information gets transferred from CAA to GAA to valine. This is the process that we call translation. So when we have a disruption or a change or a mutation in the original DNA, that information can get passed down to the mRNA and then passed down to protein. Sometimes it changes the protein, sometimes it doesn't. That's why we have different kinds of mutations. we have organized the relationship between a particular codon and the amino acid that it gets translated into in a sort of chart form, and that's what we call the genetic code. So take a second now and just kind of look at the genetic code, look at the codons, and particularly where I've circled them in green, notice that several codons can actually translate into the same amino acid, but the reverse is not possible. The way that you would read this tabular chart is that, let's just take for an example AUU, that codon. So if you look all the way on the left, you see on the left column you would find the first nucleotide, that's the A, right? Then up along the top you say this, you look at the second base or the second nucleotide. You then go to the U column. So first you go to the A row, then the U column. You come down where those two meet, and you will see each of the 4 different nucleotides then represented in that same box where you have AUU, AUC, AUA, and AUG. You then find your third codon position. You can also kind of look at the other side of the chart on the right hand side where the U is, and you come across and you find AUU, and you see that that is going to translate into an isoleucine. After you've looked at this table long enough, you will also notice that a change in the third codon position is not necessarily going to always change the amino acid. So for instance, where we were just looking AUU, if you have a mutation in that third codon position and it becomes AUC, you still get an isoleucine. If it mutates to AUA, it still gets an isoleucine. If you go down below that, GU is the same as GUC, is the same as GUA and GUG. They're all valines. Okay, so the codon position matters when you have a mutation. Many times in the third codon position, it doesn't matter. Almost always in the second codon position, it does matter, and sometimes in the first codon position, it does matter. And so if you get a mutation in the second codon position, you're going to have a change in the amino acid. If you get a change in the first codon position, sometimes you will and sometimes you won't. But because of that, different mutations, or different what we call point mutations, so changes in that base, is going to have a different effect. If you get a mutation in the third codon position and it doesn't change the amino acid, then you're not going to see any difference at the morphological level. This is kind of why we call these sort of silent mutations. Another name for a silent mutation is a synonymous substitution. Okay, so this is where there is no resulting change. The opposite of that is a non-synonymous substitution. That means that there was in fact an amino acid change. Point mutations again just alter a single base pair. We also have grouped point mutations that substitute a purine nucleotide for a purine nucleotide, or a pyrimidine for a pyrimidine as a transition, and one that substitutes a purine for a pyrimidine, or vice versa, as a transversion. If you think about it and you wonder, and you sort of hypothesize which one of these is more frequent, if you look at that chart there, the black lines indicate transition type mutations, and that sort of orange lines there represent transversions. And as you can see, there are more possibilities for a transition type mutation, but if you look at the frequency, actually transition mutations happen more frequently because you're substituting a purine for a purine, or a double ringed nitrogen containing base for a double ring nitrogen containing base, rather than, or a single ring nitrogen containing base for a single ring, as is the case with substituting a pyrimidine for a pyrimidine. And so sort of at that chemical morphological level, right, it's kind of similar, right? Whereas a transversion, it's going to cause either a little bump in the DNA molecule or a little indentation in the DNA molecule. You're changing more the general size of that place where you have your two base pairs bonding with each other. So mutation rates are important for generating genetic diversity, and so it's important for us to be able to estimate mutation rates. Traditionally, we would look for what we call loss of function mutations, so some sort of physical manifestation of a mutation, like, as is the case here, hemophilia. So we would first characterize the mutation, in this case hemophilia. There's a lack of a certain factor that's involved in the coagulation of blood and repair of wounds. And we also know that people can be carriers for it, and so it's a recessive allele. And so when we go into the general public, we look for this loss of function. We characterize hemophilia A, right? We diagnose it. We then count how many individuals are in the population. We know that the frequency of that allele, since it's a recessive allele, it has to be 2 alleles in a single individual. And so there's ways that we can estimate the frequency of that allele, and then look at it through time, and then come up with some sort of mutation rate. But if we look at this, the question would be, okay, what is the limitation of this method? So I want you to think about that for just a second. Hopefully you came up with one of the obvious issues, and that is not all mutations are going to result in a loss of function. And so it would be really nice if researchers could actually estimate mutation rates directly. That's what Deaver et al. In 2004 did when they established several independent lines of a parasite, a roundworm called C. elegans. And they grew them up under a controlled environment. And so they were able to grow them up with abundant food, no parasites, no crowding, optimum humidity, etc. C. elegans can reproduce asexually or sexually. They're hermaphroditic. And they were able to establish clonal lines, right? And so if, in fact, as they went through generations, if a mutation occurred, then they would able to, if they were able to look at the DNA, they could then detect and count all of the mutations that actually occurred, because they're coming from a single individual reproducing asexually. That's where the different lineages are coming from. So after they sampled random individuals, after lots and lots of generations, like I said, they sequenced a whole bunch of DNA. In this case, they sequenced over 10,000 base pairs, and then they were able to get a pretty good estimate of mutation rate. What resulted in this study is a mutation rate of about 2.1 mutations per individual per generation. And when they characterized those mutations, they found that about 50% of them were indels. Indels is a term we use for insertions or deletions, and so that sort of surprised the researchers. That was a little bit high, because if you think about insertions or deletions, especially when you're looking at coding sequence, inserting a nucleotide or base pair or deleting a base pair could change the frame at which those codons are read, and so that can have some major impact. They also did a follow-up experiment using mitochondrial DNA, and they found that mitochondrial DNA actually had a mutation rate about 10 times that of the nuclear genome. Others then did follow-up research, follow-up papers and things like that, comparing rates between different species of worms. So, piece by piece or group by group, we begin to get some pretty good estimates of mutation rates, and that's important for evolutionary biology, because that helps us to look at, and in an indirect way say, okay, how much potential to evolve do these populations have? In these studies, they also measured the effects of the mutations, and what they found when they looked at things like longevity, production of offspring, and other sort of aspects of fitness, they saw that over time, as the mutations accumulated in the lineages, that the fitness began to decline. And so here we have, estimates for anywhere from 0 to 250, 240 generations, and what we see is that the survival to maturity decreases and decreases, where it drops from around, in the upper 90s to the mid to slightly upper 80s. That's that slanted regression line that you see in the control, like a control should. You don't see any decrease in survivability. So the question would be, why is that? And the simplest answer is that the vast majority of mutations are deleterious. That doesn't mean all mutations are deleterious. If all mutations were deleterious, you would not have evolution occurring. But the majority of them are, and that's because there are these sort of random errors, right, in the DNA replication process. And if you're just sort of randomly, monkeying around with that stuff, you're more likely than not to cause damage rather than sort of luck upon making a change that happens to be beneficial. And here's a little exercise that demonstrates that. If you had the cover page or the inside cover page of the Origin of Species and you had one copy, and you went to the copy machine and you made a copy, and then you took the new copy and used that as the original and made another copy, every so often you would get sort of these random errors occurring, the errors in the form of these, maybe little dark spots occurring. And if you kept going generation after generation of copying, there is a chance, perhaps one of these errors makes the paper or the text in the paper, right, in that cover paper, a little bit more crisp, or might benefit it in some way. But the vast majority of those random sort of dark spots are going to make reading the paper or that page, right, even more difficult. And as you can see here, we have an example of what it looks like after 20 replications, 50 replications, and 100 replications. So just like when you are replicating a piece of paper through, iterative copying like that, mutations, for the most part, the vast majority of them would likely be, or you'd expect them to likely be, deleterious. Counteracting against that, though, is natural selection that may be selecting out the deleterious alleles. And so, for sure, and we've mentioned this before, so you don't want to take the fact that mutations are occurring randomly, and then extrapolate that out, which a lot of people have done erroneously, into thinking that, natural selection is a random process, because it's the exact opposite. It's very much a non-random process. So the follow-up question might be, okay, so then why aren't most populations getting less and less fit over time? And the answer is, obviously, that some mutations can be beneficial, but even beyond that, the vast majority of mutations aren't beneficial or deleterious, but rather neutral. So we've just looked at several types of mutations that can cause new alleles. So an allele just reminds a different version of a gene. So any change in the base pair sequence is going to produce a new allele. But now I want to look at mutations that generate entirely new genes. This is something that we call gene duplication. The most common ways in which you can produce new genes or for which you go through gene duplication is by either retrotransposition or unequal crossing over. Retrotransposition happens when mRNA or messenger RNA is reverse transcribed through reverse transcriptase, much like retroviruses do. When they invade a cell, they reverse transcribe their RNA into DNA and then insert into the genome, and this is also how this form of gene duplication occurs. The other kind is unequal crossing over. Unequal crossing over occurs during meiosis 1. If you look at the figure there, you have two chromosomes, right? They are homologous chromosomes, meaning they contain the same genes. There, the sort of figure 8 looking thing is a centromere attached to two different sister chromatids. The sister chromatids are the result of the cell going through the S phase of the cell cycle, where you synthesize DNA, and so each sister chromatid is essentially a clone of the other. And what happens is, in meiosis 1, you have chromosomes that touch at certain areas called chiasmata, and when they touch, sometimes they exchange chunks of each other. Usually they exchange equivalent chunks of each other, but every once in a while they exchange unequivalent chunks of each other. And this is what's happened here. You see on the bottom frame there the sister, or the chromosome that eventually results, the four haploid cells that result during meiosis. In this, where you see the number 2. Number 2 then has an underrepresentation of the genes. It does not contain a C gene. And the number 3 sister chromatid, which eventually becomes the new that eventually becomes the gene in the, or not gene, but the chromosome for a new gamete, then has an overrepresentation of one of those genes, the C gene. Once this happens, this is the gene duplication. You now have a gamete that has two C genes. So it'd be interesting to know just how often this occurs. And in fact, Lynch and Connery in 2000 analyzed genomes from 9 different eukaryotic organisms and found that on average, gene duplication and the subsequent increase to a high frequency happens about every 10,000 years. So, is that a lot? Is it not a lot? Not a lot. We could say that gene duplication occurs often enough to at least be an important source of genetic variation in populations. So eventually what happens is, if you duplicate an entire gene, you now have 2 copies of a gene. You only need 1 copy, and so that frees up the other copy to accumulate mutations without consequences to the phenotype. So is this a good thing? Is this a bad thing? Could you think of any advantage that this would have? And the short answer is yeah, because you can evolve new or even slightly different functions. And so it frees up all this genetic diversity, which already is kind of close to a functioning gene, but you can accumulate these mutations and in essence begin to tinker with that new gene, pseudogene, and eventually you may strike upon something that's beneficial. And the result of that is that we see gene families in lots and lots of different organisms. For example, in humans, we have the globin gene family. Here in this chart, it shows you on the y-axis the percentage of total globin synthesis, and then the different lines represents different kinds of globin. The alpha 1 and alpha 2 is there in red. You have the beta that shows low levels through gestation, and then after birth rises to a pretty high level, and then a couple of other different hemoglobins. So why, what's the purpose of this? First of all, how do we identify genes that belong to the same family? How do we say that 2 genes are genes that are two members of a gene family or just completely different independent genes. And how we identify that is we look for similarities in length and positions of the exons and the introns. Okay, the exons are the coding part of a gene, and the introns are the parts that just kind of get sliced out of them. Okay, so if we can identify, similarity in amino acid sequence, similarity in exons and introns, or positions of exons and introns, that's pretty good evidence that they belong to a gene family. So then what would the overall function of this be? And with the globin gene family, what we see is it's beneficial to have hemoglobin, different kinds of hemoglobin that have different affinity to oxygen, especially during fetal development, because if you are a fetus and your blood is being exposed and being drawn into close proximity with your mother's blood, right? It would be beneficial if you had a slightly higher affinity for oxygen than in your mother's blood. By doing that, you can draw more blood across the placenta, and with that have higher levels of oxygen, and therefore, resulting in a pretty good situation. And so we can see evidence of gene families, and we can also see in this case, some pretty good reasons why it would be beneficial to have a whole bunch of different kinds of genes. And this gene duplication helps us to understand just how these different genes may evolve. So we now have lots and lots of studies that look at gene duplications in a variety of different organisms, and what we see is the size of the families vary considerably, but that they're common. Here we just have a table that shows you the size of the gene families there all the way on the left, so it goes from 2 to above 10, more than 10 genes in a gene family. And then here we have some actual data from yeast, fruit flies, and roundworms. And what we see is that, in fact, we have lots and lots of genes that belong to gene families. For example, let's just look at fruit flies. They're in the middle. They have 404 different genes that are 404 different gene families that consist of 2 different genes. They have 52 gene families that consist of between 6 and 10 genes, and 38 that have over 10 genes in the gene family. And so this is very, very common. And it is also very, very important for evolution. And so I've now talked about a change in allele at that level, change in the gene number, and now we're going to move on to talking about a change in chromosome through what we call chromosomal mutations. A common chromosomal mutation that you probably heard about is an inversion. Just as a reminder, what an inversion is, and the diagram depicts it here, is you essentially have a chunk of the chromosome that breaks away, reorients itself, and then attaches back to the same part of the chromosome or sister chromatid. What inversions do is they cause the genes within the inversion to become linked, because post-inversion they inhibit crossing over occurring within that area, that within the inversion, and this next will show you exactly why that is. So here we have 2 homologous chromosomes. Up on top there on the left you see a normal chromosome, and then below it you see an inverted chromosome. They're homologous chromosomes, therefore they have the same genes, but as you can see in the bottom inverted chromosome, the order of those genes, EDC, has now been reversed. So when 2 homologous chromosomes come together, the like regions tend to want to synapse with each other. And so the region in both of those chromosomes that contains, for instance, the A gene is going to want to sort of synapse or pair with each other. And so what results is that if you have that inverted chromosome up there, where you, reading from left to right, go, I'll just read the gene order: A, B, E, D, C, F, then the centromere GH. On top there, it's going to synapse with the homologous chromosome, but the homologous chromosome, in order to get the similar regions close to each other, it kind of has to stretch and make sort of like a roller coaster loop underneath the inverted chromosome. And so by doing that, it now, sort of accomplishes getting like regions in close proximity to like regions on the two homologous chromosomes. So there's really no big issue with this unless you have a crossing over event occurring within that inversion. So if you do have a crossing over event occurring, say, for instance, where I have that arrow pointed, part of the inverted chromosome will then become attached to part of the normal chromosome. And so just following from left to right, starting with that inverted chromosome, with the A on the inverted chromosome, when you cross over, your new sequence of genes is going to go A, B, E, and then you have the crossing over event occur. And you will then have D, C, B, A of the normal chromosome. Likewise, and no centromere. Likewise, if you start from the right end with that inverted chromosome, the H of the inverted chromosome, you're going to have HG centromere, FCD, EF centromere, GH. So obviously this is going to cause problems in the gametes, where you have either 0 centromeres or 2 centromeres, and then you also have an over or under representation of a whole bunch of genes. This is essentially going to doom the gamete to be non-functional. So the result of that is that you simply don't see gametes that are produced with crossing over events occurring in that inverted region, and the functional sort of result of that is it locks up those allele combinations within that inverted region. So if these specific combination of alleles are adaptive or beneficial, then we can view these inversions as being sort of an adaptive result of evolution. And fortunately, there are a number of sort of natural experiments that have gone on. One of these experiments is with Drosophila subobscura. This particular species of fruit fly is native to Europe, North Africa, and the Middle East. And if you go into those different populations in Europe, North Africa, and the Middle East, what you find is that they are polymorphic for at least one inversion in 5 of their 6 chromosomes. So polymorphic meaning they have some chromosomes that have inversions and the homologous chromosome may not have inversions, or you'll see that variation. And what's interesting is the frequency of those inversions in those populations vary with latitude. So if you look at Europe, right, you look at North Africa, you look at the Middle East, and you look at the environments that you find in each of these. North Africa, obviously sort of dry, Middle East, or sort of the Levant, that sort of area, Palestine, Israel, through there, sort of Mediterranean, the southern part of Utah, sort of Mediterranean, obviously, right, environment. And then you go into North Europe and it becomes much more rainy, much more rainy in a very different environment. And so the hypothesis then is, okay, are these inversions locking up certain gene allele combination that makes them favorable for one environment or the next? And so if you track those inversions, what in fact you see is that the frequency of inversions increases the farther north you go in that population. Okay, so is that can you find causality at this point? Probably not. But fortunately, what's happened is we have had two different colonization events from Drosophila subobscura in the Americas, where we could then track what happens to those populations, and specifically what happens to the frequency of those inversions as those initial colonizing populations then spread out to other regions. And that's what the next slide shows you. So the two places that we saw these new colonizations of Drosophila subobscura in the Americas was down in Chile, in Puerto Montt, which is located about 41 degrees south latitude, and Port Townsend in the state of Washington, which is located in at about 48 degrees north latitude. In the 70s, they colonized there. We then tracked the range expansion of these flies, and then also score them or look at them and figure out the frequency of the inversions. So, for instance, if you look at the west coast of both South and North America, you see a very similar pattern. If you look at Port Townsend, Washington, those of you who've been to Washington State or British Columbia in Canada, or even up into the panhandle of Alaska, it's very wet, right? Very moist sort of environment. As you travel south, you get into Northern California, San Francisco, still kind of wet. Then you get into like the LA area and it becomes more sort of Mediterranean. Then you get down to San Diego and Baja California, and it's much more dry and desert. That's the same sort of pattern that we saw in Europe, going from Northern Europe to the Mediterranean, Middle East, and then North Africa. Same thing happens in South America. In Chile, in the southern part of Chile, Puerto Montt is about 2/3 down Chile. If you go south of Puerto Montt, you get into places like Patagonia, Chilean Patagonia. If you go north from Puerto Montt, you get into Lakes District, which is kind of wet still, up to Santiago. Santiago is Mediterranean kind of environment. North of there, the same, but then eventually you get up into northern Chile and places like the town of Arica, southern Peru, and it's dry. This is the Atacama Desert. So again, that same sort of trend. And so once you had these populations colonizing, if in fact these inversions were adaptive, then, and they were locking up genes or alleles, right? Combinations of alleles that adapted these flies to the wet environment, as the flies moved north from Puerto Montt to Santiago and then up into the Atacama Desert, you would expect that, in fact, the level of inversions or the frequency of inversions would go down. Same thing in Washington State. As they travel south into California, Central California, Southern California, Northern Baja California, where it's drier, again, you're going to see a decrease in the number of inversions. And if you look at those two graphs, that's exactly what you see. And so here, this is good proof, this is good evidence that in fact engines can serve an adaptive function by locking up those different allele combinations. These are just two pictures to give you an idea of what those environments are like. The one on the upper left-hand corner is a area that's maybe 70-80 miles north of Puerto Montt, Chile, and the one on the bottom is the Olympic Peninsula in Washington State. So as you can see, very green, very similar, very, very wet. Another kind of chromosomal mutation is what we call genome duplication. This is a large-scale mutation that results when chromosomes fail to segregate during meiosis and the production of the gametes. They then fuse together and form tetraploid organisms rather than diploid organisms. Just a reminder, a diploid organism is an organism that has two copies of each of the chromosomes. Humans are diploid organisms. This happens oftentimes in plants when in the production of those gametes you don't get the reduction from a diploid number 2n to a haploid number n, but rather you produce gametes that have both copies or both versions of every single one of your chromosomes. This happens in plants quite a bit. And as this diagram sort of shows you, so if you look there, that mental organism produces gametes that are 2n, two gametes fused together, you get a first generation of a tetra or four ploid set, ploidy means set, organism. So this individual can then self-fertilize or possibly backcross with parents or mate with siblings, right? And expose its or unite its gamete with some other gametes for producing a second generation of offspring and sort of establishing a lineage, a lineage of tetraploid plants. The big advantage of this is that you're doubling the amount of genetic material that you have, therefore really sort of ramping up the potential to evolve. This genome duplication, or the formation of these polyploidy species, can also cause a separation, obviously, from the parental population or species to the offspring population or species. And one of the reasons is that if you have a a complement of a non sort of quote unquote normal complement of chromosomes in the gametes that you produce, so in the previous slide, the 2n gamete, and you fuse with one of the other members of your species that produces gametes that just have n number, you then have 3n, or you have 3 copies of each chromosomes. When that happens, it's very hard, this triploid situation, when this triploid situation happens, it's very hard to form normal gametes, and that forms a barrier then between the new sort of lineage, the new polyploid lineage, and the previous diploid population. This slide just sort of spells out that process and the formation of a tetraploid species. Again, self-fertilization, mating with a mutant sibling, or self-fertilization or backmating with a parent are important events for this formation to occur. And we've seen this over the last 200 years occurring in lots and lots of different plants. This speciation, or this genome duplication resulting in new species. Here are just a few examples of those. If we just generally look at polyploidy in plants, we see that especially in a lot of our food crops, polyploidy is really, really common. About half of all of plant genera have some polyploid species within them, and even for the grasses, about 2/3 of all grasses are polyploid. Here you see cotton is actually a tetraploid, strawberries are an octoploid. One of the reasons that we see a lot of our food crops as that we find that a lot of our food crops are polyploid species is that a general characteristic of a polyploid is that it increases the cell size, and it also, the species, the polyploid species, also tends to be larger and robust than the ancestral diploids. But on the flip side, and we see this in agriculture a lot, sometimes they have a hard time propagating, and so you have to, in a lot of cases, have human-mediated propagation of the crops because they're polyploid. But the upside, again, is that you have larger and more robust plants, and so that's obviously a benefit to humans. Here's a nice little table that kind of sort of summarizes the things that we've talked about. I would encourage you to use this table as sort of a study guide or a study. The last section of our mutation and genetic variation chapter is to go and look at exactly how we measure genetic variation in populations. When we want to measure genetic variation in populations, we basically will score or determine the genotype of a large number of individuals in a population. The most direct way to do this is to actually go in and look at the DNA sequence. So in order to do that, or to illustrate that, we're going to go back and talk about the CCR5 gene, which is a cell receptor protein that we talked about in chapter 1 when we were looking at HIV. If you remember, there is a mutation CCR5 gene called the delta 32, which is a 32 base pair deletion. And if individuals are homozygous for that deletion, they're resistant to HIV infections, and if they're heterozygous for that deletion, meaning they have the wild type allele or version of a gene and the mutated, the delta 32 mutation, they actually don't succumb to or move into the AIDS portion of an HIV infection as quickly. So what we can do with that CCR5 gene is we can go and we can make a whole bunch of copies of those genes through a process called the polymerase chain reaction. We can amplify them, so making a whole bunch of copies of them, then expose them to a cell or to a restriction enzyme that will cut the gene, and then we can run those products on a gel, an electrophoresis gel, and we should see typical patterns emerge for individuals that are homozygous for the wild type gene, homozygous for the delta 32 mutation. The delta 32, remember, is 32 base pairs less, so they're going to have a shorter fragment. So when you run it on an electrophoresis gel, it's going to run farther down the gel than the large ones. And then we can also score individuals that are heterozygous for wild type in the delta 32 mutation, and what they should show is all of the different bands. And in that way, we can go through into a population, sample a population, and we can directly measure those genotypes, and then use those to estimate the genetic diversity of that particular population based on that gene. So this is what the molecular screen would look like that was developed by Sampson et al. Back in 1996. If you look all the way on the left, the farthest left column, you have wild type. And so in that screen what would be produced is a fragment that is, or a piece of DNA that is 735 base pairs long, and then when you expose that to the restriction enzyme, you cut it into two fragments, one fragment that is 403 base pairs long, a second fragment that is 332 base pairs long. So you add those 735 base pairs. So that would be wild type. If those were the only two bands you got on your gel, that would tell you that that individual has the wild type gene at the location of that gene on both of their chromosomes. Okay. If you look at the middle one, that's the delta 32. That you would have a fragment after exposing it to the restriction enzyme. You would have a fragment that is 371 base pairs long and 330 base pairs long. Again, notice that the smaller fragments are able to travel farther across the gel or farther down the gel because they're smaller, working their way through the agarose matrix. Okay, so that's what would that middle column is what a person homozygous for the delta 32 allele would look like. If you look at the last column on your right there, you see they have all three bands. So what that's telling you is the individual has on one of that person's chromosomes the wild type version of the gene, and on the other chromosome the delta 32 mutated version of that gene. So you can go through and you can score a whole bunch of people for these alleles, and then you can gather that data and get an estimate of the genetic diversity from it. Here, table 5.3 summarizes some of those data for different populations. You see a number of different populations from Europe, some from the Middle East and Asia, and then some from Africa. If you look at that top row, you see within the European populations, you have the Ashkenazi Jewish population. So how they do that, if you look at that next column, it shows you the number of people that they tested. Of those number of people that they tested, 26 of them were homozygous wild type, 16 of them were heterozygous individual, and a single individual in that Ashkenazi Jewish population was homozygous for the delta 32 mutation. So how you get that allele frequency that you see in those last two columns is that there's a couple of different ways of calculating it, and the book goes into the two different ways. This is the way that I think is the easiest, is to kind of envision that population of 43 individuals is really a population of 86 alleles, because each individual has two alleles at that particular gene. And so if you sort of think about it that way, you have a total population of a total allele population of 86 alleles. Of that total population, you have 26 individuals that have two copies of that wild type allele, right? So that 26 you would times by 2, that equals 52. And then in the heterozygous population, you have 16 individuals that have just one copy of the wild type gene. So then you add 52 plus 6, that equals 68. Divide that by 86, the total number of alleles in that population, and you get 79.1%. You can do the same thing with the delta 32. So in the delta 32 population, you would have 16 individuals that have just one copy of that delta 32, so you have 16 of those alleles, plus you have one individual that has two delta 32 alleles, so you would add those two alleles to that 16 and get 18, divide by 86, and you will get just over 20%. And so those are your two allele frequencies. You have your wild type at just under 80%, and your delta 32 just over 32%. You can go through and do that same thing with the Icelandic population, the British population, Italian, all the way through. If you look down at the Yemeni population in the Middle East, you see that there are 34 individuals, and 34 individuals are homozygous for the wild type. Therefore, that frequency, the frequency of the wild type allele in that population would be 1 or 100, right? And the frequency of the delta 32 would be 0. You can go through, use this table and go through and calculate those allele frequencies. The other statistic or measure that you can calculate is the number of heterozygous individuals in that population. So if you look at the Ashkenazi Jewish population, you see that 16 individuals of the 43 are heterozygous heterozygous individuals. If you divide those 16 individuals by the total number of people in that population that were scored, you have 16 divided by 43, which is about 37%. So 37% of the population is heterozygous at this particular gene. You could then go through and measure a whole bunch of different genes in that Ashkenazi Jewish population and get an estimate for how much what we call mean heterozygosity exists in that population. So these are all ways of measuring the genetic diversity of a population. This next slide we'll talk more specifically about these measures. So this measure that we call mean heterozygosity can be interpreted in two ways. One is the average frequency of heterozygotes across loci. The other way, sort of the flip side, just a kind of another way of looking at this is the fraction of the genes that are heterozygous in the genotype of an average person. Okay, so the average person out there, what percentage of their genes are they on average going to be heterozygous? So a population that does not have a lot of genetic diversity is going to have a lower number, a lower mean heterozygosity. For example, cheetah populations that have gone through a bottleneck, right, have been reduced, dramatically reduced, a couple 10 to 15,000 years ago, and there's not a lot of genetic diversity at those alleles. They're going to have a low mean heterozygosity. If you take an individual cheetah, right, so describing that population, they're going to be a very sort of homozygous, right, at their alleles. And then if you look at any particular gene or loci, right, in that population, you're going to see that a very low number, a few of those genes or few of those loci are going to be heterozygous. And so that's two different those are just two different perspectives on mean heterozygosity. Another type of statistic that we look at is what we call the percent of polymorphic genes, and that's just the fraction of genes in a population that has at least two alleles. Okay, so these are diversity summary statistics that biologists use. So to illustrate the difference between these two measures, we could have a fictitious population of 100 individuals where we're going to measure 100 different genes in that population. When we actually go in and measure those genes, we find that all 100 of them are polymorphic within that population, meaning that we don't have any of the genes that only have one allele. They all have two or more alleles. But we might find that at each of those 100 genes, the mutant allele is rare, say, occurs at 1% of the time. In that case, what would happen is we would have if we measured the percent of polymorphic loci, we would say, okay, all 100 of them, 100% of them are polymorphic, but the mean heterozygosity would only be 1%, meaning if you were to sample a single individual from that population and measured all 100 genes, right, you'd probably only find one of those genes that individual was polymorphic or heterozygous at. So that kind of illustrates the difference between those two different measures. They aren't, a lot of times students sort of think, aren't these the same thing, when in fact they're not. And this is a good example of a case or a scenario where they would not be the same. When we have actual studies that go out there and measure the genetic variation, initially we sort of thought that in fact we probably wouldn't find a lot of genetic variation, but when we would actually go out there and measure it, we found that in most populations lots of alleles are usually present at every gene in the genome. This is a figure taken from the book. On the top, it's the one that's not labeled there is for vertebrates. The middle one is invertebrates. The bottom one is plant, and that just shows you the mean heterozygosity in a whole bunch of different samples. So what we found, kind of to our surprise, is that in fact there are lots of different alleles, not just one most advantageous allele at a particular population. We have also looked at when looking at particular genes, in fact, like the gene for cystic fibrosis, we found that there were lots and lots and lots of different mutations that caused cystic fibrosis, and there wasn't just one mutation that caused fibrosis, but there were 500 different mutations, each of which is a different allele. And so what we're finding is that at lots of genes we have lots of different variation. In order to explain that variation, we had to change our thinking from just like an allele that was highly deleterious as contrasted with an allele that is highly advantageous, but more thinking about lots of alleles that aren't advantageous or that are sort of what we call neutral. In the next slide we talk a little bit about that. So the two major hypotheses in explaining the existence of this high amount of genetic diversity is the neutral hypothesis, which says that most mutations are kind of selectively and functionally equivalent, and they're maintained by drift. Some mutations arise and they drift up or they drift down to extinction. But on average, what you're seeing is you see that, in fact, not they aren't deleterious enough to necessarily make it so they get weeded out of the populations, and they're not advantageous enough that they're going to go to fixation at 100% of the population. So there's sort of this that's what we sort of know as the neutral hypothesis. The selectionist hypothesis says that selection is actually favoring rare individuals, the heterozygous, and that different alleles at different times and places will be selected for. So that's what's maintained that that genetic diversity. And here is presented sort of as two very distinct sort of hypotheses. Probably in real life, there's a little bit of both of those going on to maintain the genetic diversity. So that's the end of this section, Chapter 5. Hopefully we now have a better understanding of mutation as the ultimate source of genetic variation, and how genetic variation is, or the importance of genetic variation in a population for evolution, and how we actually go about measuring that genetic variation.